Hello, this is Kranti, and in this podcast, we will be discussing the NEET essentials of human digestive system. But before we go ahead, actually, what is meant by digestion? Digestion is a complex catabolic process where complex non-diffusible biomolecules are hydrolyzed into simple, absorbable, diffusible molecules, where the complex food is hydrolyzed or broken down into simple, diffusible molecules with the help of enzymes. And these enzymes all function in the presence of water, so they come under the class hydrolases, because in enzymes we have different classes of enzymes like oxidoreductases, transferases, ligases, isomerases, hydrolases. So these enzymes in digestive system come under hydrolases because they function in the presence of water. And the complete human digestive system can be studied under two divisions: the alimentary canal and the digestive glands. Okay, so in alimentary canal, what is alimentary canal? It's a one long continuous tube which starts from mouth to anus, and it's about 8 to 10 meters long. It is about 8 to 10 meters long. And the other thing is the digestive glands. Okay, I forgot to write the digestive glands. So there are different digestive glands. In the mouth, first you have the salivary glands, and then in the esophagus you don't have any salivary glands, don't have any digestive glands. And then in the stomach you have gastric glands, and just near the stomach you have the liver, gallbladder, pancreas, and finally in the intestine you have the intestinal glands. Okay, these are the digestive glands. First we'll discuss the various structures which come across the alimentary canal, and then we'll discuss the digestive glands. First, coming to alimentary canal, the first part is the buccopharyngeal cavity, which is just in front of the esophagus. This buccopharyngeal cavity has two parts, mouth and pharynx. And again, mouth has two parts, vestibule and buccal cavity. So what is vestibule? Vestibule is this slit. So mouth, it is the opening between the two lips. And what is vestibule? Vestibule is a space between the lips and the teeth. It's not so well developed in human beings, but in herbivores like cows and rabbits, they take the bits of grass and keep it in between the lips and the teeth. This space is called as the vestibule. It's not so well developed in human beings, but that vestibule will continue inside into the buccal cavity. So this vestibule and buccal cavity, also called as oral cavity, is called the mouth. So vestibule, it is the space between the lips, the cheeks, and inside the teeth. And buccal cavity is the inside oral cavity in which the food is placed. And this buccal cavity, it is bound on the top. It's bound on the top by palate. On top you have the palate, and on the bottom you have the tongue, and to the sides you have the teeth. Okay, so on the top you have palate, and the bottom you have the tongue, and to the sides you have teeth. So the top, bottom, and sides, it's bound, the buccal cavity is bound by these three structures. Okay, first let us see how it looks like. So this is the palate, this is the nasal cavity, and this is the tongue, this is the teeth, and this is the oral cavity or the buccal cavity. So this is the palate, and this palate is of two types. The palate is two types. In the front you have, this is the hard bony palate, the anterior or the front bony palate, and the posterior soft palate. Okay, the front part is the bony palate, and the back side you have soft palate. And this bony palate here, if you see, there are some lines here. Here there are some lines, some ridges. They are called as palatine rugae. Palatine rugae. And this palatine rugae, they give some friction to hold the food while chewing, because when the tongue is mixing the food with the saliva, or when the tongue is pushing the food onto the teeth while mastication, the food may slip here and there. But this palatine rugae, they hold the food so that it doesn't slip here and there. It gives some grip. And at the back, this is the soft palate, but this soft palate has a small hanging flap like structure. This is called as uvula. Okay, and these are the internal nares, and this is the larynx or the voice box, and backside you have this food pipe. So the food comes from oral cavity into pharynx, and also the air also comes from nasal cavity into pharynx. We'll discuss about pharynx later. But this food, when you're swallowing, when it's coming from oral cavity into pharynx, there is a danger of this food slipping back into the nasal cavity. But that danger is prevented by this uvula, because when you're swallowing, this uvula will rise up like this. It will rise up and prevents this backward flow into the nasal cavity, and the food will only come into the food pipe. There is also danger of the food going into this larynx, but you have here the epiglottis. The epiglottis closes the glottis, and the uvula closes the internal nares. So the only chance for the food is to go from oral cavity to pharynx and pharynx to food pipe. So uvula, it prevents the backward flow of food from oral cavity into nasal cavity. That is about palate. Now next, coming to the tongue. Tongue is a muscular protrusible organ. It has a lot of functions. The first function, it is taste. The tongue also helps in speech. It helps in degglutination, that is swallowing. It helps in pushing the food onto the teeth. It helps in mixing with the saliva, and in some animals like dogs, cats, it helps in ingestion of the food. So there are different functions of tongue. But this tongue, it's held to the floor of the oral cavity by a fold. That's called as frenulum. The tongue is held with the floor of the oral cavity by a fold. That's called as frenulum. And the surface of this tongue has many taste buds. But these taste buds are grouped into some groups. These groups are called as papillae, taste papillae. So the taste papillae are group of taste buds. And this taste papillae, there are 4 types. First is the filiform papillae, and the fungiform papillae, and you have next foliate papillae and circumvallate papillae. Filiform papillae is a conical shape. It's conical shape, and the fungiform is mushroom shaped. The foliate is something like leaf like, and the circumvallate is knob like. Okay, now remember, these 3 type of papillae, they have taste buds. They have taste buds. But this filiform papillae, they have no taste buds. But then what is their function? They help in breaking the food particles into smaller particles. They help in breaking the food into smaller particles. So these are the 4 types of papillae. But remember, for NEET point of view, the only papillae which do not have taste buds is the filiform papillae. But fungiform, foliate, circumvallate, they all have taste buds. They don't have taste buds, and these 3 have taste buds. Now coming to the structure of each taste bud. See, if you see, each taste bud is like this, conical, and at the tip you have an opening which is called the taste pore, and this is the taste cavity, and the taste cavity is lined by some cells which are called the gustatory receptors, because gustation meaning taste, so they perceive the taste. And every gustatory receptor has some cilia, some hair like structures. They are called as gustatory cilia. So when the food is placed in the mouth, it's mixed with saliva, it's converted into a solution, and that food in the form of solution goes into the taste bud, into the taste cavity, and this cilia will detect that solution. If it is it will detect the solution, and it will produce a small electric impulse that goes to the gustatory nerve, and the gustatory nerve goes to the taste center of the cerebrum. So in this way you have the structure of taste bud. And coming to the types of taste buds, as there are 4 types of taste, you have 4 types of taste buds. four types of taste buds. At the tip you have the sweet taste buds, and behind the sweet taste buds you have the salt taste buds, and to the sides you have the sour taste buds, and at the base or at the bottom of the tongue you have the bitter taste buds. But remember, sweet taste buds, they are due to molecules like glycols and aldehydes, sweet taste. And salt taste is because of anions of some ionizable salt. Anions of ionizable salt will give you the salt taste, because if the salt cannot be ionized, that doesn't give any saltish taste. So the salt should it should actually be dissociable. It should be ionized. Only then you will get that salt taste. And sour taste is because of organic acids, and bitter taste is because of alkaloids like caffeine, nimbin, like that. They give the bitter taste. Alkaloids give the bitter taste. Alkaloids are the nitrogen containing organic compounds. They give the bitter taste. And remember, to perceive any taste, the food should actually mix with saliva. If the tongue is dry, and if a dry food is placed on the tongue, it doesn't give any taste, because the food should mix with saliva, convert into solution form. Only then the gustatory cilia will perceive or detect that solution and produce that particular taste. That's why if you put some salt on your dry tongue, doesn't give any taste. So there are four types of taste. 1, 2, 3, 4. There is another type of taste: hot taste, spicy taste. But is that any taste? Hot taste, spicy taste? That's not a taste, because the cool taste of mint or the hot spicy taste of chilies is actually not a taste. It's only the irritation of the taste buds, the irritation of the gustatory cilia. That irritation is felt as a burning sensation or the coolness of mint, but that's actually not a true taste. And sometimes we get a white coating on the tongue. When you sleep for the overnight and when you wake up in the morning, you get some white coating. Why do you get the white coating on the tongue? That's called as coated tongue, white coating. That's because the saliva contains calcium hydroxide. So when you breathe in air, definitely there is some air in the oral cavity also, and that contains about 0.03% carbon dioxide. So that calcium hydroxide, which is in the saliva, will mix with carbon dioxide and forms a calcium carbonate deposit that will sediment on the tongue. This calcium carbonate will sediment on the tongue, and that's a nice perfect breeding place for the bacteria. So that calcium carbonate deposit on the tongue will give you that coated tongue. That's about tongue. And next, coming to teeth. Teeth are the most strongest structures in the body. They're stronger than the bone, and the human teeth are thecodont, they are diphyodont, and they are also thecodont, diphyodont, and heterodont. What do you mean by thecodont? The teeth are deeply rooted, deeply embedded in the sockets of the jaw bones, deeply rooted. You have some teeth like acrodont, which are the tip of the jaws, like in snakes, like the fangs, they're acrodont. But thecodont are deeply rooted in the jaw bones. And diphyodont, di meaning two. There are two sets of teeth arising in the life of any human being. Two sets of teeth. One set is the milk teeth, and the other is the permanent teeth. Milk teeth, and the other is the permanent teeth. Milk teeth is also called as deciduous teeth or temporary teeth, because they only start from about 6 months of age and stay up till 6 years of age. But from 6 years, the teeth fall off and get replaced with permanent teeth, also called as successional teeth, because they are the successors of the milk teeth. Okay, now these milk teeth are very small, they are very weak, but permanent teeth are large and strong. Okay, they are small, weak, but permanent teeth are big and strong. When you look at the dental formula, for the milk teeth it is 2, 1, 0, 2. Okay, I'll write like this: incisors, canines, premolars, molars. What is dental formula? Dental formula is the formula showing the number of teeth in one half of upper and lower jaw. One half of upper and lower jaw. So totally you have 2, 3, 4, 5, 5 and 5, 10. 10 on the top, 5 on the upper jaw, 5 on the lower jaw, and this side 5 and this side 5. Totally you have 20 teeth. Because remember, there are no premolars in milk teeth. But permanent teeth dental formula is 2, 1, 2, 3, or incisors 2 by 2, canines 1 by 1, premolars 2 by 2, and molars 3 by 3. So totally you have 32 teeth, 16 in upper jaw and 16 in lower jaw, 32 teeth. And these milk teeth, they generally start erupting from the jaw bones at 6 months of age, and they will completely, all the 20 teeth will form by after 2 years of age. And once all the 20 teeth are formed by 2 years of age, they will stay up till a maximum of 6 years of age, and from 6 years they slowly fall off one by one, and slowly get replaced with a stronger one, permanent teeth. And they will almost form by 18 years of age, except the last wisdom teeth. But the last wisdom teeth, by 18 years of age you are 2, 1, 2, 2, but by after 24 years of age you get even the last wisdom teeth also. So that's why in the mother, she stops feeding the child with the breast milk by about 6 months of age. Slowly she gets weaned out, and that's why from that age they will develop teeth and they start feeding on solid food. So okay, that's milk teeth. And heterodont, definitely I told you just now, heterodont are the teeth with different shapes, different sizes, different functions. Like you have the incisors, canines, premolars, molars. So different types of teeth. In some animals you have all the same type of teeth, which are called homodont, but in human beings it's heterodont. And coming to the components of the teeth, if you look at inside, okay, so this is the outermost part, which is called the enamel, the outer exposed bluish white shining part, that's the enamel. And this is the dentine. It's made up of dentinal tubules, so enamel, this is the dentine, and you have here this gums, and this is the cement. So this is the cement. So it has 3 parts: enamel, dentine, cement. This is the hard tissue. See, because let me erase this. If you look at the components of teeth, the teeth has 2 components: the hard tissue and the soft tissue. Okay, the teeth has 2 components. One is the hard tissue, and the other is the soft tissue. The hard tissue includes these 3 parts: the enamel, dentine, and cement. Whereas the soft tissue includes the innermost one, which is the innermost is the pulp cavity. This is the pulp cavity, and the outermost one, this is the gums. Gums are also called as periodontal membrane. So the pulp cavity and gums or periodontal membrane. Okay, so hard tissue are the hard parts: enamel, dentine, cement. So enamel is the hardest part, the bluish white exposed part. What you see is the enamel. It's very strong because it's made up of calcium and fluorine, which give it a lot of strength, and it is regenerable. As the enamel is lost, new enamel can be produced, but it's non-living. But the dentine is living. Why? Because it's made up of dentinal tubules. So if you look at each dentinal tubule, it's like this. This is a dentinal tubule, and here you have a blood vessel which supplies blood capillaries, Okay, an artery which gives arterioles, a vein which gives venules, and also a nerve which gives nerve fibers. So these blood capillaries and these nerve fibers, they also extend into these dentinal tubules. They extend into the dentinal tubules. And these dentinal tubules are lined by some cells. These cells are called as odontoblasts. Odontoblasts. These odontoblasts can produce any lost enamel. Okay, they can produce, if enamel is lost because of friction or abrasion or any acids, these odontoblasts produce the calcium and the minerals, and the enamel can be repaired or regenerated whenever there is lost. So you have some living cells lined in these dentinal tubules. This is the dentinal tubule. So in this dentinal tubule you have the blood capillaries and also nerve fibers. That's why when you try to scratch this dentine, there is no sensation. Sorry, the enamel, there is no sensation. Why? Because it's non-living. But when there is an injury to the dentine, definitely there is a pain. Sometimes because of infections, because of some bacteria like Streptococcus mutans, which grow in between the teeth, because when there is no good oral hygiene, for example, when you don't brush your teeth twice a day, especially during the night time, there may be some food particles stuck between the teeth, and that food particles will attract bacteria, and the main bacteria is the Streptococcus mutans. This is a bacteria which grows in between the teeth, and that releases lactic acid. Throughout the night when you sleep, the bacteria nicely thrives in between the teeth and produce lactic acid, and the lactic acid, since it's an acid, it erodes this enamel, causing small tiny microscopic pits. But when there is no proper care taken to the teeth, these pits may deepen, they may deepen and they may deepen, and they may reach up till the dentine. That's called as pulpitis or dental caries. And whenever you eat any hot food or a cold food or a spicy food, that will enter through these pulp cavities. They may enter into the odontoblasts and cause irritation of these nerve fibers. Okay, sometimes these cavities may even bore the dentine and also reach up till the pulp cavity, which causes a very severe pain when anything goes up till the pulp cavity. Okay, that's called sensitive teeth or dental caries, which is the main cause is the lactic acid produced by Streptococcus mutans. That's why you have to brush your teeth with any alkaline material. That is why our toothpastes are alkaline in nature, because they can neutralize these lactic acids in between the teeth. And they also say that you have to brush it's good if your toothpaste has a lot of salt, because that salt can kill the bacteria in between the teeth, because salt kills bacteria by exoosmosis. They lose water, the bacteria lose water, and they get dehydrated, and the bacteria will die. That's good method of maintaining your oral hygiene or the health of the teeth, by brushing your teeth with either alkaline material or any salt. That's about teeth. Next, coming to pharynx. Pharynx, I already told you, it's a muscular passage. For example, this is a mouth, this is the palate, this is the uvula, this is the oral cavity, this is the nasal cavity, this is the internal nares, and you have this is the larynx, and this is here our foot pipe. This is the pharynx. This is the pharynx. So pharynx is the common passage for both food and air. Okay, for example, the food from oral cavity comes into pharynx, and the air from nasal cavity also comes into pharynx. But the food from pharynx goes into food pipe, whereas the air from pharynx goes into larynx. The entrance of larynx is called glottis, which is covered by epiglottis, but the entrance of food pipe is called as gullet. Okay, So the food goes from oral cavity to pharynx and pharynx to gullet or into the food pipe. From here you have the esophagus or the food pipe. That is pharynx. So pharynx is just a common muscular passage, common for both food and air. The only place where both food and air meet is the pharynx. The food doesn't go into nasal cavity because of uvula. The food doesn't go into larynx because of epiglottis. That is about pharynx. Now let us look at the next structure, esophagus. Esophagus is commonly called as food pipe, and it's about 25 centimeters long, and there is no digestive enzymes produced in esophagus. Its only job is to help in pushing the food down into the food pipe. The food is converted into food bolus, and the esophagus shows a series of wave-like contractions and relaxations which are called as peristalsis. Peristalsis, because when the food pipe gets pressed here, the food bolus comes down, and again when the food bolus comes here, the food pipe gets pressed over here, again it comes down. So this part again goes back to the normal position and the food bolus comes here. Now the food pipe will press over here. Okay, so and that way it keeps moving down. This is called as peristalsis. So esophagus doesn't produce any digestive enzymes, but there is some little digestion occurring because of the saliva mixed with the food. Saliva contains salivary amylase, which starts digestion of carbohydrates. So there is digestion of carbohydrates occurring in the food pipe, but that's because of the enzymes produced in the saliva, present in the saliva. Here there is no enzymes produced by the esophagus. Esophagus job is only meant to pass the food pipe from pharynx to stomach with the help of wave-like contractions and relaxations called as peristalsis. Sometimes if the stomach is filled with overloaded with food, or with too much fat food, oil food, or there may be some indigestion, and such food is again thrown back from the stomach through food pipe, pharynx, oral cavity, and out of the mouth. That is called as vomiting or reverse peristalsis. Reverse peristalsis or vomiting, it commonly occurs when the food pipe has a lot of substance which is allergic, or then there is food poisoning, or when there is indigestion. Because sometimes, some people, I've seen my friends, they during a birthday party, they eat a lot of cakes, a lot of cream. The cream is a lot of fat. It doesn't get digested. You just try to drink a spoon of oil, will it get digested? You get vomiting. So similarly, if the stomach is loaded with too much of fats which cannot be digested, or if there is any food poisoning, or any allergic food, or any food which you don't like, that may trigger reverse peristalsis, vomiting, through which the food will come out of the mouth. So that's about esophagus. And now we will discuss about the stomach, the big one and the most important one. Stomach is a muscular J-shaped bag-like structure. So this is the food pipe, and this is the diaphragm. The food pipe starts from the neck. Food pipe passes through the neck, passes through the chest, and then there is a diaphragm. It will pierce the diaphragm and enter into the abdomen. So the chest region, this is the abdomen region, and the organs of chest region and organs of abdomen region are separated by a muscular dome-shaped partition, which is called as a diaphragm. So this food pipe, it pierces the diaphragm, goes into the abdomen, and leads into a J-shaped elastic muscular bag-like structure, which is called the stomach. And the esophagus enters into stomach through an opening called as cardiac sphincter. Cardiac sphincter, or it's also called as gastroesophageal sphincter. And there's another sphincter here which is called pyloric sphincter. Okay, food from esophagus comes into stomach through cardiac sphincter, and the digested food or the partially digested food from stomach goes into the duodenum through pyloric sphincter. These sphincters, they are circular muscles. They're always closed. Only when the arrival of food bolus they will open and come here, and only after the digested food is digested, the pyloric sphincter will open, and then the food will come into the duodenum. Okay, and this stomach, let me just show you the stomach in bigger. So look at this is the stomach. So the stomach has three portions. This hunch-like part where the food is temporarily stored, that is called as a fundic stomach. Okay, this is the fundic stomach, and like this you take this is the cardiac stomach, and this is the pyloric stomach, and you have the cardiac sphincter and the pyloric sphincter, and the inner lining of the stomach, which is called the mucosa, has digestive glands which produce digestive juice. We'll discuss about gastric glands later. And this is the cardiac sphincter, pyloric sphincter, and this stomach. Now, so stomach is to which side, left side or right side? If you look at here like this, it's to the left side. It's to the left side, and just below the diaphragm. If you look at this, is the diaphragm. Just below the diaphragm, to the left side, you have the stomach. Okay, you have here your lungs. Left side. So when sometimes some people, when they overload their stomach with too much food, the stomach expands and they get suffocation. Why? Because when the stomach expands like this, it pushes these lower lobes of the left lung, this fundic part, it pushes the diaphragm, and the diaphragm pushes the left lung, which causes less volume, less lung capacity, and that will increase the rate of breathing. It causes suffocation, because sometimes when you overload your stomach with too much food, you get suffocation. That's because this fundic stomach, it goes up pushing the diaphragm, and the diaphragm pushes the lungs, and there is a less lung capacity, which increases the rate of breathing. So that's like that. And next, coming to the intestine. In intestine you have small intestine and large intestine. Small intestine you have duodenum, jejunum, ileum, large intestine, cecum, colon, rectum. The small intestine is about 6.25 meters long. This is just about 1.5 meter long. Okay, so let's look at once again. This is the esophagus, and this is the stomach. So the first part of this is the cardiac sphincter and pyloric sphincter, and this pyloric sphincter will enter into a C-shaped part. The first part of small intestine, which is the duodenum. Duodenum is about 25 centimeters long. Jejunum is about 2.5 meters long, and the ileum is 3.5 meters long. Okay, so the first part of small intestine is this C-shaped part, which is called the duodenum, and that will lead into loosely coiled part, which is jejunum. Okay, where almost the complete digestion is finished, and absorption of food starts in ileum, and ileum is a much coiled part. It's much coiled part. Jejunum is loosely coiled, and ileum is very much coiled, because when it's very much coiled, the length increases, and the more digested food is absorbed. Rate of absorption will be increased. So loosely coiled and much coiled ileum, and that leads into the first part of large intestine, which is the cecum. You have the vermiform appendix, the cecum, and this is the colon, and this is a rectum, and we'll discuss the large intestine later. But I just want to concentrate on this duodenum here. To the right side of the duodenum, you have liver, and this liver has two lobes, the right lobe and the left lobe. The right lobe gives rise to right hepatic duct. The left lobe gives rise to left hepatic duct. Okay, I'll show this in a different picture. Sorry. So this is the food pipe. So this is the liver. It has two lobes, the right lobe and the left. So the bottom part of right side is the right lobe and this is the left lobe. The two lobes are connected by a ligament. That ligament is called as falciform ligament. Falciform ligament. And the right lobe gives rise to right hepatic duct. The left lobe gives rise to left hepatic duct, and they both join to form the common hepatic duct. So right hepatic duct, this is the right hepatic duct, this is the left hepatic duct, and this is the common hepatic duct. The common hepatic duct joins with cystic duct. This is the cystic duct. At the end of cystic duct you have the gallbladder. The common hepatic duct and the cystic duct both join to form what is called as a bile duct. This is the bile duct, and that bile duct will join with the pancreatic duct. This is the pancreatic duct, at the end of which you have the pancreas, and the bile duct and the pancreatic duct, pancreatic duct both will join to form what is called the hepatopancreatic duct. Hepatopancreatic duct. And that hepatopancreatic duct, it opens into a small sac. A small sac that is called as the ampulla of Vater. Ampulla of Vater. And ampulla of Vater finally opens into the duodenum through an opening which is called the sphincter of Oddi. Sphincter of Oddi. So once again have a look over here. Right lobe gives rise to right hepatic duct. Left lobe gives rise to left hepatic duct. Both join to form the common hepatic duct. The common hepatic duct joins with the cystic duct. Common hepatic duct, cystic duct both join to form the bile duct. And the bile duct joins with the pancreatic duct. Bile duct, pancreatic duct both join to form the hepatopancreatic duct. That opens into what is called here a temporary reservoir of bile juice and pancreatic juice, that is the ampulla of Vater. And finally that ampulla of Vater opens into duodenum through a small opening called sphincter of Oddi. Sphincter of Oddi. So remember, what is ampulla of Vater? Ampulla of Vater is a sac. Whenever the bile juice comes into the ampulla of Vater, it is temporarily stored there. Only upon the arrival of the food, the acidic chyme, when it comes into duodenum, only then the ampulla of Vater will release the bile juice or it will release the pancreatic juice. So ampulla of Vater is a temporary reservoir, a temporary storehouse of bile juice and pancreatic juice that is poured into the duodenum through this sphincter of Oddi. That's about the first part, duodenum. And jejunum, I said it's a loosely coiled part, and ileum is a much coiled part, the longest part of small intestine, the 3.5 meters long. And then okay, so I'm erasing this. So now this is again the stomach, esophagus, stomach, and this is the duodenum, and this is the jejunum, and then this is the ileum, and finally you enter into the cecum. Cecum is the first part, the starting part of large intestine. It goes into the colon, and in colon you have the ascending colon. This is the ascending colon. This is the transverse colon which runs from the liver side to the pancreas side, and then you have the descending colon, and then the oblique one is sigmoid colon, and then you have the rectum, and then this is anal canal, and this is the anus. So this is cecum. Cecum at the bottom, it gives rise to some small tube about 8 to 10 centimeters long. That's called the vermiform appendix. Cecum, and this ascending colon, transverse colon, descending colon. So this is the ascending colon, this is the ascending colon, transverse colon, descending colon. This is a sigmoid colon, and this is the anal canal, and this is the anus, and this is the rectum. So here you can see the three parts, cecum, and this is the colon, okay, and this is the rectum, three parts. So first coming with cecum, the starting part of large intestine. See, look at here, let me show you this part in a bigger view. This is cecum, and this is the appendix, vermiform appendix, because it looks like a worm. This is the last part of the ileum. This ileum, it actually first opens into a small sac-like structure. This sac-like structure is called as sacculus rotundus. saculus rotundus, where the undigested food is temporarily stored here. Only when it is filled, it will be disposed into the cecum. Okay, cecum, it harbors some symbiotic microorganisms which release cellulase enzyme, but it's not so well developed in human beings. We treat it as cecum can be treated as a vestigial organ in human beings, but in herbivores whose primary diet is cellulose, because no, in animal kingdom no animal can produce cellulase enzyme, only bacteria or microorganisms can produce cellulase enzyme. But since herbivores main diet is cellulose, the cecum will harbor some symbiotic microorganisms which produce cellulase enzyme, and that will digest the cellulose in the cecum. And then you have the colon. The colon is drawn into some pouches. These pouches are called as haustra. What is the meaning of these sacs, the saculated, these pouches are called as haustra. What is the function of haustra? See, because when the undigested waste material, when it comes into cecum, it's with large amount of water, but we cannot afford to lose so much of amount of water. It should be reabsorbed again. So as this undigested food moves front, the colon absorbs water. The main job of colon is to absorb water from the excretory material, from the fecal material, from the undigested waste. But if it's straight and continuous like the small intestine, it may easily come out and the water absorption may be less. But because of these pouches, the undigested food material will stop here and there. It will stop here for some time and the water is absorbed. It's pushed forward. Again the water is absorbed. It will more solidify and it's pushed forward. Again it will stop in this haustra and water is absorbed. It will more solidify. So it stops at each haustra for some time, so that there is sufficient time for the absorption of water, during which the excreted material will get more solidified, and finally it's temporarily stored in the rectum. Rectum is a temporary storehouse of all the undigested waste material. Only when it is filled, then you get the ease of defecation, and then there is a internal anal sphincter and external anal sphincter. First the internal anal sphincter will open and then it comes into anal canal, and then the external anal sphincter will open and then it will come out defecation. So that is the alimentary canal. Okay, and next we will discuss about the digestive glands. Digestive glands. First coming to salivary glands. Salivary glands are the first digestive glands, and there are three types of salivary glands: the parotid glands, and next the submaxillary or submandibular glands, and lastly sublingual glands. Sublingual. Parotid glands are the largest in size and they are just 2 in number. Submaxillary are small in size, they are also 2 in number, but sublingual are the smallest and they are many in number. And where are the parotid glands found? Parotid glands are found below the auditory canal. Okay, it's below the auditory canal, below the ear or below the external auditory canal you find the parotid glands, and they contribute to about 25% of the saliva. Whereas submaxillary glands, they are found below the angles of the lower jaw. Here you find here one and here one, and they contribute to 5% of the saliva. But sublingual glands, there are many tiny glands which are below the tongue. Below the tongue you have many salivary glands, sublingual glands. They are smallest and they contribute to remaining 70% of the saliva. So parotid glands are the largest, they contribute to 25%. They are below the auditory canal. Sometimes these parotid glands may have a painful viral infection which is called mumps. It's a painful swelling of the parotid glands because of the infection of myxovirus parotiditis. That virus, it causes infection of the parotid glands. It will swell, a painful swelling, and then it will be there will be a painful pain in opening the mouth, or pain in the swallowing, or it may even lead to some earache and fever, headache, all the symptoms of mumps, viral infection of parotid glands. But anyhow, what is present in saliva? Saliva has 99% of water plus salts. These salts can be like sodium salts, potassium salts, chloride salts, bicarbonate salts, like this. And the remaining 1% is different ingredients, like you have IgA antibodies, the same antibodies found in the human colostrum and the mother's breast milk. You have the IgA antibodies, very powerful antibodies. It can kill a variety of strains of bacteria. Okay, this IgA antibody breaks the bond between the NAM and NAG of the bacterial cell wall. The bacterial cell wall is made up of peptidoglycan, which are alternating NAM and NAG, N-acetylmuramic acid and N-acetylglutamic acid. For example, this is NAM, NAG, NAM, NAG, like this. So you have a bond which is called the beta-1,4 glycosidic linkage. This IgA antibodies, they can go and attack bacteria. They can attack the bacteria and attack this NAM and NAG, and they can disrupt it. And also you have one more enzyme called Lysozyme. It's an antibacterial enzyme. This will break these bonds. This beta-1,4 glycosidic linkage, it will break these bonds. Lysozyme will break the beta-1,4 glycosidic linkage between these monomers of peptidoglycan. Hence the cell wall is disrupted, or the bacteria will burst. Anyhow, you have the IgA antibodies, Lysozyme, and there is also a lot of mucus, and you also have enzymes, like you have salivary amylase, and a very little of salivary lipase, lipase. And what is salivary amylase? What does it do? It will break the carbohydrates, or it will break the starch, into maltose. Okay, the salivary amylase, in the olden days it was called initially as ptyalin. It's a starch splitting enzyme. It breaks starch into maltose. So the first nutrient which starts digestion is starch. The first nutrient in the food which starts digestion in the mouth is starch. About 30% of starch is digested here itself. Okay, 30% of starch in the food is because of the digestion from the salivary amylase. 30% of starch in the food is digested because of the salivary amylase, ptyalin. So that is about saliva. Next, coming to gastric glands. Look at here. This is the stomach, and the stomach it has the mucosal lining, and here and there some group of cells, these mucosal cells, they will act as gastric glands. They release gastric juice, hence they are called as gastric glands. This mucosal epithelium of the stomach will release some enzymes, hence they are called as gastric glands. And these gastric glands, I forgot to say the pH of saliva. pH of saliva is about 7.4 pH, alkaline pH, slightly alkaline pH, 7.4. But the pH of gastric juice is 6.8, and liver pH is 7.8. Initially it will be 8.8, but that is reduced to 7.8. The bile juice pH is 8.8, but the bile juice, after going to gallbladder, it reduces the pH. This is the bile juice pH 8.8, and this is the gallbladder bile pH 7.8. Anyhow, so this is the gastric glands, and these gastric glands will release gastric juice. Okay, this gastric juice has different enzymes, but before going to that, I want to say that in this gastric glands there are three types of cells. Okay, what are the three types of cells in the gastric glands? You have first the chief cells. Chief cells are also called as peptic cells, and then the other one is the parietal cells. Parietal cells are also called as oxyntic cells, and lastly coming to the goblet cells, or the cells which release the mucus, goblet cells or mucus cells. Now coming to the chief cells, in the gastric glands, they release pepsinogen, they release prorenin, and some enzymes like gastric amylase and gastric lipase. This all you find in this pepsinogen. In this chief cells we release all these enzymes, whereas the parietal cells or oxyntic cells, they release HCl and Cassell's intrinsic factor, whereas this goblet cells they release mucus and bicarbonates. Now this HCl will maintain an acidic pH, very strong pH of about I'm sorry, here it's not 6.8, it's 1.8. okay, HCl pH is 1.8. now this pepsinogen, it's an inactive form of pepsin. So immediately after pepsinogen is released, it's activated by HCl into pepsin, and the prorenin also converts gets converted to renin. So pepsinogen is an inactive enzyme. It has about 12 amino acids, but the last 2 amino acids are cut off by the HCl, so that becomes a 10 amino acid pepsin, we call decapeptide, which is more exposed, because pepsinogen, it has 12 amino acids and it's folded. The tertiary structure, it's folded. The last 11th and 12th amino acids, they cover the active site, but when the HCl cuts off the last 2 amino acids, the active site is now exposed. That is called as pepsin, a decapeptide. And similarly prorenin also is activated to renin by HCl. What is the function of pepsin? Pepsin, it will break proteins. It will break proteins into peptides and proteoses. Means smaller segments of proteins. Long proteins are broken down to small chains, which are called peptides or proteoses. And what is the function of renin? Prorenin is released only in children up till about 2 years of age, or up to maximum up till 3-4 years of age. Why? Because in them the main primary food is milk. Milk is the main diet, and milk is a liquid, so it can pass off through the stomach fastly, and the calcium and the casein proteins are not properly absorbed. But when the prorenin converted to renin, this renin will convert the milk, it will coagulate the milk into a semi-solid form, and when the milk is in a semi-solid form, it will slowly pass through the stomach, and all the benefits from that milk can be obtained, like all the calcium, all the proteins, all the fat, everything can be absorbed properly. So renin helps in coagulation of milk, but the amount of prorenin release will gradually decrease after 3 years or 4 years of age, and finally stop by 6 years of age. And gastric amylase, it breaks carbohydrates. It breaks carbohydrates to disaccharides, and gastric lipase will break the fats into diglycerides. And HCl, HCl, it activates pepsinogen, it activates prorenin, and HCl, it will maintain acidic atmosphere in the stomach, so that any acid sensitive bacteria are killed in the stomach. And Cassell's intrinsic factor, it helps in absorption of vitamin B12, cyanocobalamin, and vitamin B12 is essential for the formation of red blood cells. So with deficiency of vitamin B12 causes pernicious anemia. Okay, so when there is no, Cassell's intrinsic factor is just like a carrier carrier molecule. It carries vitamin B12 from the food and dumps it into the blood. But when there is no causal intrinsic factor, definitely that causes deficiency of vitamin B12. And deficiency of vitamin B12 causes a pernicious anemia, where there is decrease in RBC count. So you should understand what is the difference between anemia and pernicious anemia. Both anemia and pernicious anemia is because the symptom is a decrease in RBC count. See, because for the formation of red blood cells, at one stage you require iron for the formation of the hemoglobin, and for the manufacture of this structure you require vitamin B12. The manufacturing of RBC structure, you require vitamin B12, and iron is required for hemoglobin. So when there is deficiency of iron and therefore no RBCs, that's called as anemia. But when there is no B12 and therefore no RBCs, that's called as pernicious anemia. So anemia and pernicious anemia both are resulting in a decrease in RBC count, but anemia is due to deficiency of iron and pernicious anemia is due to deficiency of vitamin B12. And the deficiency of vitamin B12 can be due to food deficiency. The food may not have, not food, because vitamin B12 is the only vitamin which is produced by intestinal bacteria. But if that bacteria are harmed because of antibiotics or any hot food, the bacteria, that useful bacteria also get killed, and therefore no vitamin B12. Or though if they produce vitamin B12, if there is no sufficient causal intrinsic factor, then also it leads to deficiency vitamin B12, which causes pernicious anemia. And finally, coming to goblet cells, which release mucus and bicarbonates. Remember here, the pH of the stomach of the HCl in the stomach is very high, very acidic, 1.8. Such a strong pH can even digest the stomach itself. But the inner lining of this mucosal lining of the stomach is protected by the mucus and the bicarbonates. So mucus and bicarbonates, they protect the stomach wall from the HCl. They protect it from acidity, because mucus and bicarbonates, they both are alkaline. They protect the stomach from the acidic action of HCl. That is about the gastric glands. Now we will discuss about liver. Liver, to which side you have liver, left or right side? It is to the right side. See, look at here. This is the food pipe, and this is the stomach, this is the duodenum, and here you have the liver. All the structures, left hepatic duct, right hepatic duct, and the common hepatic duct, the cystic duct, gallbladder, and the bile duct, and this is the pancreatic duct, and this is the pancreas, and the hepatopancreatic duct, and you have the ampulla of Vater and the sphincter of Oddi. Okay, now as the liver has two lobes, the right lobe and the left lobe, and the two lobes are connected by falciform ligament, I said. So, but each lobe is further made up of tiny microscopic hexagonal units. They are called as lobules. There are many such lobules. Each liver has about 1 million lobules. We also call it as hepatic lobules. And these hepatic lobules, there are about 1 million in each in the liver. Each lobe is divided into the tiny microscopic hexagonal structures like this. Okay, this is called as hepatic lobules, and the hepatic lobule is covered and protected by a thin connective tissue which is called as a Glisson's capsule. Each hepatic lobule is covered by a thin connective tissue which is called as Glisson's capsule. At the center you have a central venule, and at each edge you have a hepatic triad which is made up of 3 blood vessels, and the cells in this lobule are called as hepatocytes, hepatic cells or hepatocytes. But these cells are not completely distributed. They are found like strands. Like this, like columns. These are a bunch of cells like this, and these are some bunch of cells like this, like strands like this. Okay, these are the hepatocytes, and between each strand of hepatocytes you have an empty space where blood is flowing. That is called as hepatic sinusoids. hepatic sinus or hepatic sinusoids, where you can see the blood flowing. And very important, in this hepatic sinusoids you have some cells which are phagocytic in nature. These are called as Kupffer cells. They are called as Kupffer cells. They kill any bacteria which are in the blood which is coming into the liver. Kupffer cells are some same like white blood cells. They wander in the hepatic sinusoids. They are phagocytic in nature. They kill any bacteria which come into the liver. That's about a liver. Now coming to this, liver releases the digestive juice which is called as bile juice. Bile juice. Okay, and this bile juice, you know, it will it's about it's alkaline. I told you. See, the bile juice which is coming from the liver is having a pH of 8.8, but after it goes into the gallbladder, the gallbladder function is to remove water, concentrate it, so it becomes it becomes less alkaline. So gallbladder, it takes the bile juice, concentrates the bile juice and gives it again. So this is called as bile juice, and it's called as gall juice. So the bile juice pH is about 8.8, but which is coming from the gallbladder, this is 7.8. The bile juice coming from liver is 8.8, but that coming from the gallbladder is 7.8. So overall it's alkaline. And this bile juice, it has different components, like it has bile salts, it has bile pigments, like that. And the bile salts are like sodium carbonate, sodium bicarbonate, sodium taurocholate, sodium glycocholate. These are all bile salts. And what is the function of the bile salts? They help in emulsification of fats, because you know, when the food coming from the stomach, when it comes into the duodenum, first the liver will release bile juice, and then the pancreas will release pancreatic juice, because only when the liver is mixed, when the bile juice is mixed with the food, only then the fats in that food can be digested upon the action of the pancreatic juice. Okay, just like you know, when there is an oil to your hands, when you've eaten some biryani or any fried food, there's a lot of oil and dalda and ghee onto your hands. If you directly wash it under water, the oil will not go away from your hands. So what you will do, you will first apply some soap, and then it can be cleaned with water. So the soap is also some sodium salts which remove this oil. So here also that's also emulsification. The same thing happens here. The bile juice, which contains all these bile salts, they mix with the fats and oils and lipids, reduce their surface tension, break into smaller molecules, and that is called as emulsification. Breaking large fats into small molecules is called emulsification, and that is done by the bile salts in the bile juice. And bile pigments, there are two types of bile pigments: bilirubin and biliverdin. Bilirubin is a yellow color pigment, which is the main component of bile juice. About 70% of the bile pigments is bilirubin, and remaining 20 to 30% is biliverdin, which is pale green in color. From where did these bile pigments come into bile juice? You know, the liver is the graveyard of red blood cells. I'll show it here. See, for example, let's imagine this is the bone marrow. This is the bone marrow, and the red blood cells are born from the bone marrow, erythropoiesis. This is the red blood cell, and each red blood cell, it lives for 120 days. After 120 days of lifespan, every red blood cell, it becomes old, and it becomes inefficient in carrying oxygen and carbon dioxide. Such old red blood cells are sent to the liver, and they are destroyed over there. Daily about 10 to the power of red blood cells are destroyed per day. That's why liver is called as a graveyard of red blood cells. But when these red blood cells are destroyed, the hemoglobin pigment is also destroyed. Okay, here the hemoglobin is destroyed. The hemoglobin is broken down into two components, the heme and the globin, because hemoglobin has two components. heme, which is the iron containing inorganic part, and the globin is the protein containing organic part. The heme will return back to the bone marrow for fresh synthesis of hemoglobin, whereas the globin will remain in the liver itself and further break down into these bile pigments, bilirubin and bilirubinin. The globin, which is the protein part, will remain in the liver and break into further small pigments, bilirubin, bilirubinin. They're poisonous, they're harmful. Then you might ask me a question: if this bilirubin and bilirubinin are coming along with the bile juice and mixing with the food, isn't it not dangerous because it's mixing with the food? Though the bilirubin, bilirubinin are mixed with the food, in the villi, the bilirubin and bilirubinin are not absorbed into the blood. Only the digested food will be absorbed by the villi, but the bilirubin, bilirubinin will be left unabsorbed and it will move along with the feces. That is why the fecal matter is yellow colored, because of this bilirubin, which comes from the breakdown of hemoglobin. Okay, so that is a function of liver. Liver has a lot of functions. Liver, Greek word, it's a helper, meaning it helps everywhere. It has about more than about 3000 functions. It has a lot of functions, like it helps in deamination, detoxification. When some people consume a lot of tobacco, lot of alcohol, that nicotine in tobacco or the alcohol in the tobacco, in that ethanol in that alcohol will damage the liver and cause liver cirrhosis, or the liver may not remove the bilirubin and bilirubinin. It will be in the blood itself, in the liver itself. That may lead to jaundice also. If the bilirubin, bilirubinin are not excreted out, and if they remain in the blood, or if they remain in the liver, the liver becomes very sensitive and tender. It may swell, causing liver cirrhosis or jaundice. That's all because of various hepatotoxins or hepatitis virus which damage the liver. Next, coming to pancreas. Gallbladder, I already told you, its only one function is to concentrate the bile juice. Now let us look at it. This is the food pipe. Sorry, I'm drawing so many times. So this is the liver, and this is the pancreas. Now, one more thing I forgot to tell you. Liver is the largest gland in our body. It's about 1.2 to 1.5 kg. The liver is about 1.2 to 1.5 kg, largest gland in our body. The second largest gland is the pancreas. The smallest gland in our body is the endocrine gland, pituitary gland. So this is now the pancreas. Pancreas is also called as exoendocrine gland or endoexocrine gland, because it has both exocrine portion and endocrine portion. Or it's also called as a mixed gland, because it is the only gland which can release enzymes and hormones. Enzymes, that's why we call exocrine, and hormones, that's why we call endocrine. And it's also called as a complete gland, because pancreas is the only digestive gland whose juice, the pancreatic juice, has enzyme necessary to digest all the nutrients. The pancreatic juice has enzymes necessary to digest proteins, carbohydrates, lipids, everything. It can digest all the nutrients in the food. That's why it's called as a complete digestive gland. But liver is the only digestive gland which does not release any digestive enzymes. They will give you in the NEET exam. The only digestive gland which does not produce any digestive enzymes is liver. It only releases bile juice, which contains bile salts that will help in emulsification of fats, so that they can be easily digested by the pancreatic lipases. So now coming to this liver, the exocrine part is represented by SNI cells which release the pancreatic juice. The endocrine part is represented by pancreatic islets, or the islets of Langerhans, which release two hormones. Pancreatic islets, they release two hormones. One is the insulin and the other is glucagon. So when there is increase in glucose levels, that is converted to glycogen by the action of insulin, and when there is decreasing decreasing glucose levels, the glycogen is again converted to glucose by glucagon. So increasing glucose levels will signal the pancreas to release insulin, and that extra glucose will be converted to glycogen, and the glycogen is stored in the liver and the skeletal muscles. But when there is a decrease in glucose levels, like during long traveling or during fasting or starvation, or forcibly when you don't eat, what happens is there is decrease in glucose levels. That will signal the pancreas to release another hormone which is called glucagon. That glucagon will go and act on liver and skeletal muscles. It brings all the stored glycogen back into the blood and breaks into glucose. Okay, that's why since here glycogen is being produced, we call it glycogenesis, and here since it's breaking down, it's called glycogenolysis. Conversion to glucose to glycogen is called glycogenesis because it's being produced. Genesis. This is glycogenesis and this is glycogenolysis. Okay, we'll talk about that in endocrine system. Now coming to the pancreatic juice, which is prepared by the SNI cells. Pancreatic juice. So I'll erase this one. Maybe I may draw it again. So the pancreatic juice. Pancreatic juice, I told you, it contains enzyme necessary to digest all the nutrients. It contains proteases which digest the proteins. It has amylases which digest the carbohydrates. It has lipases which digest the lipids. All these are present in the pancreas. And it also has nucleases. All these are present in the pancreatic juice. But remember, these proteases are in inactive form, which we call proenzymes. Means they are produced in inactive form. Like for example, you may have trypsinogen, chymotrypsinogen, and procarboxypeptidases. All these enzymes are in active form. Only, so this is the food pipe, stomach, and the duodenum. So this is the bile duct and the pancreas. So remember, only after the pancreatic juice comes here, the duodenum releases one enzyme called enterokinase that will activate trypsinogen to trypsin. The mucosal epithelium of the duodenum, which has one cells called Pallet cells, which we'll discuss later, that will release one very important enzyme, enterokinase. So what will enterokinase do? Enterokinase will first activate trypsinogen to trypsin, and then trypsin will convert this chymotrypsinogen to chymotrypsin, and also it also converts the procarboxypeptidases to carboxypeptidases. So remember once again, the pancreatic juice has these three proteases, all are in inactive form, which we call proenzymes. After the trypsinogen comes here, then the mucosal epithelium of this duodenum, which has some cells called Pallet cells, they release enterokinase. Okay, these cells will release enterokinase. We call that as Pallet cells, which are the cells of the intestinal glands found in the duodenum. We'll discuss it later. The Pallet cells will release enterokinase, and the enterokinase will immediately break trypsinogen to trypsin, chymotrypsinogen to chymotrypsin, procarboxypeptidases to carboxypeptidases. Now, what will these three enzymes do now? Whether it's trypsin or chymotrypsin and carboxypeptidases, these three will convert all the substances like proteoses, peptides, and dipeptides. It will break all this into dipeptides. Okay, which one? This trypsin, chymotrypsin, this trypsin, chymotrypsin, and carboxypeptidase. These three will break the proteins, proteoses, peptides into dipeptides. They break into dipeptides. And what will this amylase do? This amylase breaks carbohydrates to disaccharides, this amylase. What will lipase do? It breaks fats into diglycerides, this lipases. And what will nucleases do? It will break the nucleic acids like DNA or RNA is first broken down into nucleotides, and then a phosphate is removed from it to form the nucleosides. That is done by this nucleases. So finally, what's happening in the duodenum is the proteins are being converted to dipeptides, the carbohydrates are converted to disaccharides, the fats have been broken into diglycerides, and the nucleic acids have been converted to nucleosides. This has been completed now. Now, the job of intestinal glands. So I just wanted to keep it here, and I'll erase this to show the intestinal glands. Okay, now coming to the intestinal glands. The intestinal glands are found in the mucosal epithelium of the small intestine, and these intestinal glands are two types. You have the crypts of Lieberkühn, LEI, and the Brunner's glands, So I forgot to say about villi. We'll discuss. See, villi, you know, the wall, the inner wall of the small intestine, whether it's duodenum, jejunum, and ileum, the inner wall is thrown into some foldings, which finger-like foldings, which are called as villi. When you look at the anatomy of anatomy of small intestine, it has an outer thin coat which is called the serous layer, and inside to it you have what is called the muscular layer, and then you have the submucosa, and inside you have mucosa. So serous layer is the outside, middle muscular layer, and submucosa and mucosa. And again, in this muscular layer you have outer longitudinal muscles and inner circular muscles. Outer longitudinal muscles, inner circular muscles. So just remember, outside you have the serous layer, and then you have the muscular layer, and then you have the submucosa and the innermost mucosa. In the second layer, muscular layer, you have outer longitudinal layer and inner circular layer. Sometimes there may be even oblique muscles also, like in stomach, because the stomach should contract longitudinally, horizontally, obliquely. It should contract in all directions to mix with the gastric juice. So sometimes, like in stomach, you have even oblique muscles. Okay, longitudinal muscles, circular muscles, and oblique muscles. Now coming to the structure of villi, I told you the inner wall, the inner mucosa of small intestine is drawn into some finger-like foldings like this. This is the mucus layer. These are the mucus layer cells, which these cells are called as enterocytes. They are called as enterocytes. And here you have an artery which gives arterioles and capillaries. You have a vein which gives venules and capillaries. You have a nerve which gives nerve fibers. And also there is one lacteal. There is a lymph duct, which I'll show it green. So let us think this is the lymph duct, and this lymph duct gives one lacteal. All these three they make a, so this is the mucosa, and this three is the submucosa, and then you have the muscular layer, and outside you have the serous layer. Okay, you have the serous layer and the muscular layer. In this muscular you have outer longitudinal muscles and inner circular muscles, and then you have the submucous layer which has all these blood capillaries and lymph vessels. And this is the villi. Inside villi, what do you find? You find the blood capillaries of arteries, veins. You find nerve fibers, and you have one large lacteal, and there is also some connective tissue. All these are found inside the villi. But here and there some enterocytes. Now all enterocytes they have their inner wall, the inner cell membrane is drawn to some foldings which are called microvilli. Okay, if you look at this particular cell, it's like this. The inside cell membrane, which is to the lumen side, the cell membrane is drawn to some foldings which are called as microvilli. So this is the villi, and this is the microvilli. This microvilli, when you look at the surface, it looks like the bristles of a brush, very soft, and it has a velvety texture, very soft. That's why it's called as a brush border. The border of villi is called brush border because of these microvilli. Okay, but here and there are some cells which are called goblet cells. For example, this is a goblet cell, this is a goblet cell, this is a goblet cell, and this is a goblet cells. They will release mucus. So the cells lining the villi are called enterocytes. Here and there are some enterocytes which are called goblet cells. They release mucus. Okay, because this mucus it has to reduce the acidic chyme. I forgot to say one point. The partially digested food in the stomach which is coming from the pyloric sphincter is called as chyme. It's acidic in nature. So when the acidic chyme comes from the pyloric sphincter into duodenum, that acidity is neutralized by the mucus released by the goblet cells. Okay, the acidic chyme which is coming from the stomach through the pyloric sphincter into the duodenum is neutralized by the mucus produced by the goblet cells. Okay, now let us think there is one more villi like this, and the space between these two villi is called as crypts of Lieberkühn. This is another villi. So one villi, another villi, another villi. The space between these villi, the long deep pits found between the villi, okay, the space between these villi are called as crypts of Lieberkühn. And those crypts of Lieberkühn, they have two types of cells. Okay, so if you look at here, there are some cells here. There are two types of cells in this crypts of Lieberkühn. One is the Paneth cells, and the other is the Argentifin cells. One is Paneth cells and the other is the Argentifin cells. These Paneth cells will release the enterokinase. So only after the acidic chyme, after coming into the duodenum, the liver will release bile juice, it gets mixed with it, emulsification, and then the pancreas will release pancreatic juice. The pancreatic juice has trypsinogen, chymotrypsinogen, but they will not immediately start their action. Only after the Paneth cells release the enterokinase, and after they are activated into trypsin, chymotrypsin, and carboxypeptidase, then they will start their action in the duodenum. And what is their action? Converting all the proteins, proteases, peptides into dipeptides. That happens here. Paneth cells. And also Paneth cells also release several other enzymes also. There are several other enzymes. Paneth cells release enterokinase and also release other enzymes. Like to digest these dipeptides, it releases dipeptidases, and to further digest the disaccharides, it releases disaccharidases. Okay, and to digest this diglycerides, it releases lipases, and to digest these nucleosides, it will release nucleosidases. So that this will mark the complete digestion of the food. because finally this Pancreas cells will release all these enzymes. So this enzyme, they'll finish the digestion, means they'll completely break down dipeptides into amino acids, disaccharides into sugars, and this diglycerides into fatty acids and glycerols, and nucleosides are also digested. Okay, and then this argintifin cells will release hormones. It will release two hormones. One is secretin hormone and the other is serotonin. Serotonin is a very powerful hormone. It's a very powerful stimulant of the small intestine. It stimulates the contraction of the small intestine. It helps in the peristaltic movements of the small intestine. Okay, serotonin hormone, it stimulates the muscular contraction of the intestine, which allows the peristaltic movements of the small intestine, and therefore push the digested food forward. The digested food is pushed forward across the small intestine because of peristalsis, and that peristalsis is because of the serotonin hormone. And as the food is being pushed forward, it keeps on being absorbed and absorbed and absorbed by the villi. Finally, it's left with the undigested food in the ileum, which releases into the cecum. So these two are hormones, and the Brunner's glands they will release mucus, and this will also release one more thing. It also releases lysozyme. Okay, Pancreas cells release enterokinase. They release these enzymes, and also an antibacterial enzyme, lysozyme. Once again, I'm telling you, Pancreas cells will release enterokinase, some digestive enzymes, and even lysozyme, which is an antibacterial enzyme. And argintifin cells, hormones like secretin and serotonin. And Brunner's glands will produce mucus. But one important point, these Brunner's glands are found only in the duodenum. This is important for NEET. These are found only in duodenum. The Brunner's glands are found only in the duodenum. Okay, and Pancreas cells are also found in the duodenum. But argintifin cells, they are found throughout duodenum, jejunum, and ileum. It's found duodenum, jejunum, and ileum, everywhere. And finally, the last important point, the complete mixture of all these things is called as succus entericus. Succus entericus are also called as intestinal juice. I mean, the secretions of Pancreas cells, secretions of argintifin cells, and this mucus, all together, this complete mixture produced by the intestinal glands is called as succus entericus or intestinal juice. The complete, everything, these hormones, these enzymes, the antibacterial enzyme, these hormones, this mucus, everything together is called as succus entericus or intestinal juice. And finally, the food is completely digested and it's absorbed by the villi into the blood. And that marks the end of digestion. Okay, and that's it. I hope it's helpful. Thank you.